When you think of microbes, what do you normally think of? What comes to mind? For most people, it's probably disease, the mold growing on your bread at home. Maybe it's the microbes that we use to make yogurt or cheese. Well, for our guests today, they're not like most people. So when they think of microbes, they think of the brilliant red of Serratia marcescens. They think of the glowing neon blue of bioluminescent dinoflagellates, the kaleidoscope of colors that E. coli can be made to express. So we're going to have a really interesting conversation with them today about how the beauty and intrigue of microbes can be used to open up a world of microbiology to a broad audience through education and outreach. So with us today, we have Jeff Morris, who's an associate professor at the University of Alabama, Birmingham. We have Sarah Adkins, who's a graduate student working with Jeff Morris and a recipient of the NSF Graduate Research Fellowship. And we have Katherine Lontok, who's ASM's public outreach manager. So let's start off just by having you all tell us, how did you become interested in microbial art? How did this become a thing that you started pursuing?
Okay, so I was a biology student and I got sidetracked and also became an art student. So at UAB, I guess I was in art school while I was also growing frustrated with a lot of the ways that my science courses were being taught. And then I stumbled into Dr. Morris's Intro to Microbiology course, and one thing led to another, and I wound up in his lab. And I think both of us realized how cool the Agar Art contest was, and we both sort of saw it at different times, and we were like, we can do something with this, and we can make this into a teaching exercise. So I piloted a teaching exercise using a few students, like many people in this room have done, like using it as an outreach activity, but we wanted to not only use it as an activity, but convert its potential into a way to get students interested about science and see the microbial ecology that were happening on the plates, to have them create their own experiments. So we transformed the activity into a course-based undergraduate research experience, and that is also what I study for my dissertation.
Really interesting. So Jeff, how did you go down this path initially?
Well, for me, I was a new assistant professor, and I was trying to do some cool new things with this class, this intro micro class that I was teaching. And I just happened to get an undergrad working in the lab who happened to be a double major in biology and art. At the same time, I'm seeing the ASM Agri Art contest. I'm thinking about these things. I'm always trying to figure out how to leverage all the stuff around me into cool new projects, and it was just serendipity, basically. So we just kind of riffed on the idea of using art in the class until something happened with it.
Oh, very cool. How about you, Katherine?
When I came to ASM as a staff member, it was a program that had been started by my predecessor, sort of on the fly, like, let's just see what happens, right? And so I came in and had to start running the second year of this contest, and was just amazed at the entries that we got. And the fact that this contest, unlike some contests that combine science and art, I'm thinking specifically of like the Nikon Small World contest, you don't need like a ton of expertise to do this, or super fancy equipment, right? I don't want to spoil things, but you'll see later on that even kids can produce something really cool. So yeah, it spoke to my heart as a public outreach person, basically.
So we're going to come back to that, and we'll actually look at a few examples of the amazing entries to the Agri Art Competition. Before we do that, Sarah, I want to ask you, you mentioned that your thesis work is kind of looking at this art and microbiology interface. What are you doing for your thesis?
Yeah, I don't know. So I am an artist. I do a lot of performance art, ceramics, and photography, sort of new media. And now I'm a bench scientist, and now I'm funded to do education work. So I think my dissertation sort of tries to pull all of those three things together. So we do educational assessments on this art cure that we've made. So I was talking about how the students in the class make the agar art, and it informs them to make their own experimentation. And we think that's really cool, right? Like everyone here is probably like, yeah, that's super cool, but how do we know that it works? And so that's where I started to explore education research and use like validated instruments and focus groups to compare student attitudes in that class versus student attitudes in a cookbook microbiology class, where they would go through the lab out of a traditional manual. And we saw that the students in our class, according to the interviews and the survey instruments, identified as scientists more, and some of the research that came out of last semester showed that they liked the agar art more than anything else they did in the semester that we looked into. So my dissertation is sort of like looking at the avenues that agar art can be used through education research, through my own bioartistry, and also how it can inform like the science that we're doing at our university.
Wow. Okay. So while we're talking about agar art and how it impacts attitudes towards microbiology, Katherine, I want to ask you about ASM's Agri Art Competition. You mentioned it was kind of formed on the fly, and I think it's a very popular activity now. How many entries do you get, and where do they come from?
They come from all over the world. I believe we had 26 countries represented this year. The number of entries kind of varies. We had more than 100 this year. We had maybe 170. I actually, rather than just the number, I've been looking sort of at the quality. You know, if you follow the contest over time, the entries have just gotten astronomically more detailed and impressive and just mind-blowing. So that's been really interesting to see. We have a huge following on social media for these photos. You know, people who get drawn into our account through these awesome images that get shared all over the place. And I get probably at least an email every couple weeks from someone who's like, hey, can I use this in my newsletter? We want to publish this in our magazine, and that sort of thing. So it's really great to see that spread.
Yeah, and you can see all these entries on ASM's Facebook page, and what I really love is looking at the comments on the Agri Art submission, because there are people who write in and they say, what is this that I'm looking at? And they come for the art and they learn something about science in the process. And it's just really
that's my mom.
Oh, that's your mom. All right. That's great. I love seeing that kind of aha process,
Right.
that people go through. So speaking of the Agri Art Competition, we're going to look at a couple of examples. So here on the screen over here, you can see an example. This was the first place winner of the Agri Art Competition this year. It's titled The Battle of Winter and Spring, and it was submitted by an undergraduate student in Tbilisi, Georgia.
Yes.
Speaking of international entries. So this Agri Art depicts, I think it's very cool. It depicts winter on the left in white, and the bacteria that make up the winter half of this plate are kind of retreating from these red flowers made of Serratia marcescens. And that happens because the Serratia are secreting antibiotics that kill the white bacteria. I thought this was a really cool entry, A, because it's just beautiful to look at, but B, because this is really something that demonstrates that uses the function of bacteria in the artistic message of the piece. And it really got me, it's a fascinating idea. It got me wondering, how can we use, art, microbial art, to kind of demonstrate some aspects of bacteria beyond just their physical beauty? Maybe Jeff and Sarah, do you want to talk about this one?
Yeah, so this is interesting because in our class, this is one of the things that students sort of get out of their agar art more than just about anything else. So what we have the students do is they isolate soil microbes, right? They select them based on what the colonies look like, and then they make art. But the thing is, they're not like skilled artists like this, right? So really, what they end up doing is looking at the art and trying to figure out why it doesn't look like what they thought it was going to look like, right? And so in what we have them do, they do all this other stuff too to characterize the microbes, and we have them try and synthesize all of that together to try and figure out what their ecology is like, right? And they often intuit that they're producing antibiotics, or that they're competing or even cooperating in different kinds of ways. And then the last few weeks of the semester, they develop their own experiments that they can do using the equipment that we have in the lab, and they actually execute those to try and test some of these ideas. But it's often based on something like antibiotics or direct sort of microbial competition kinds of things.
That's very cool. So we actually have a couple of examples of works that have come across from your students' work. So what are we looking at here?
So this is Chromobacter violaceum, which is a very striking purple bacterium. We always offer the students a couple of sort of classic microbes like this, so like Chromobacter and Serratia and some other things. And they always gravitate towards the purple. They love the purple. But the thing is that it doesn't reliably make the purple on different kinds of media or in different kinds of conditions. So what you're looking at here is this student made a caduceus, right? It's a medical student or a pre-med student. And you can see that it produces like almost a black pigment on the BHI plate on the right, but it kind of erratically produces the color on the left. And what you don't know is that there's other microbes on here as well. And you can see that the presence of the snake microbe in here seems to be altering the pigment production of the Chromobacterium on the left plate, which led them sort of to think about why that is and to work on that.
Very cool. And I think we have one more example of a. All right. So this is I think this is a Jiff. So this should play, hopefully. There we go. All right. So what's happening here, Sarah?
Yeah, so similar thing, that the same design is painted on two different dishes, and this is a time lapse. So there's, I think, day 0, day 1, maybe 3 days out, and a week later. And you can see how, even though it's the same image using the same bacteria, they create drastically different results depending on the media. So you can see on the left that the Chromobacter, I don't remember what the white one was, but they're sort of like melding together in ways they don't do on the right. And so we think that these types of explorations and the things that you can use bioart or agar art for aren't just limited to aesthetic objects, but can really sort of tease out scientific, interesting facts about the microbes or the things that students or other people are working with.
So one thing I just want to say about this is that the really cool thing about this, in my opinion, is that the students aren't going out there with the idea of testing a particular thing. It's just free play, right? So it really has the potential to generate like real eureka moments, right? I mean, they could come up with anything, who knows, right? And they certainly have. I mean, they found things I didn't even know existed. They found situations where non-modal bacteria will stick to modal bacteria and swim with them. Right? I didn't know that was a thing, and I looked it up. Apparently, there's a literature on bacterial hitchhiking, but still, it's fascinating the kinds of things they stumble across using these plates.
Very cool. So we have another Agri Art entry I want to show now. So this was a 3rd place winner this year, titled Sustenance. And I think, Katherine, do you want to tell us some about this piece?
Sure. So this is actually a piece that was produced by a scientist-artist collaboration. So Mehmet Berkman is a research scientist at New England Biolabs, and he's worked for several years, actually, with a mixed media artist named Maria Panil-Cobo. And they've had a very fruitful collaboration. And basically, he offers his lab to her, right, and has taught her how to culture bacteria, how to identify them, and then she has created these amazing pieces of art using these bacteria. And they actually also won the 2015 Agri Art contest. They were the 1st place winners. That was the first one that we ever did. And they are featured in the how to make agar art on our YouTube page if you want to look at that as well. So it's fascinating. They've had a really interesting collaboration, and Mehmet is a great guy to talk to. He very much does not believe in separating biology, science, and art. He doesn't understand why there's such a divide in the public eye about these things, because to him they are very much in concert with each other. So
So Catherine, can you tell us a little bit about what we're looking at in this particular image?
Sure. So what we're looking at, we've got two plates here, and it's become almost a sculpture. I don't know if the artist would call it that. And linked with this red yarn. So the plate depicting the breast here has several different species on it, including a species that was actually directly cultured from Maria's breast. So she is a new mom. She's got a 1-year-old. It's her first kid. And so that was sort of the inspiration for this piece. And then in the plate depicting the fetus, she's used some bacteria that were actually collected from her daughter's hands, right? So she's really incorporated the whole message of the piece into how it is created. It's very cool.
So this piece quite literally depicts the microbial connection between mother and child.
Yeah.
It's
Yep.
pretty amazing.
Mm-hmm.
So, all right, so we've heard examples now, some really interesting examples of where we've gotten these microbes from, right? So in that image, microbes were isolated from the breast of a mother and the hand of a child.
Yes.
Jeff, you've discussed isolating microbes from soil and using them in art. What are some of the places, where do all these microbes come from? That we're seeing all these beautiful colors, some glowing microbes, some colorful microbes. Where are they coming from?
Well, yeah, the first time I ever did this by myself, I found Chromobacter in the garden outside of our building. So, and that's the really sharp purple one.
Right.
So they're everywhere.
So one thing I always like to point out to the students, which I think kind of blows their minds a little bit, is we look at these things aesthetically, right? We look at the pigments and we think that the purpose is that they're colored, but they evolved before anything had eyes, right? So these pigments are older than the perception of color or image or anything like that. So they do other things, right? And trying to figure out all the different tasks that these pigments accomplish for bacteria. I mean, there's all sorts of stuff. They're antibiotics, they're photosynthetic, antioxidants are pretty common, right? So there's all sorts of different things that they do. So often, I mean, the fact that these things have color is directly related to their physiology and how they behave on the plates.
Right.
And are most of these bacteria used in art, are they mostly natural isolates, or are we looking at a lot of engineered bacteria that produce colors?
Yeah. So we see actually in the Agri Art, we see a mix. Usually the pieces that we get from professional microbiologists usually use naturally occurring colorful bacteria. We've also started hosting these public Agri Art workshops in partnership with community labs. And very often, for that purpose, we use transgenic or organisms that have genes introduced into them to make them colorful, right? So like E. coli with GFP, for example. And the reason for that is really it's just about a level of expertise that you need to manipulate these things. So it's much easier to have E. coli that are expressing several different colors, right? Because they'll all probably grow at about the same rate than it is to use naturally occurring colorful organisms where you have to kind of time things just right. And, one thing might interact in an unexpected way if you're not super familiar with the organisms, right? So yeah.
So it's interesting. So more goes into this than just painting colorful microbes on a plate, right?
Yes.
There's a lot to consider.
Yeah, I mean, so I said earlier that you can create something beautiful even if you're not an expert microbiologist, and that is definitely true. However, right, some of the things, for example, that Maria is creating with Mehmet, they've been working together for years, right? She actually has become, I would say, an expert over that time, right? She doesn't necessarily have formal schooling in microbiology, but she knows the organisms that she's working with, right? And she knows exactly how to manipulate them and how they're going to grow next to each other so that she can get those really intricate, detailed images.
Wow. All right. So some of you may have noticed we've got here on the screen now, we've got some kids winners. So on the left here, the image on the left is a picture of a butterfly. It was created by a young microbiologist, a talented young microbiologist named Kate Lynn, who's 10 years old. Blew my mind when I found that out, right?
Yeah.
This is an amazing picture. Who are these talented young microbiologists? Where are they coming from, and,
Right.
and how do they get the resources to do this?
Right. So all of these kids created their agar art in one of these public agar art workshops. So one of the things that we've been expanding sort of year after year as we do this contest is these partnerships with community labs. And the reason that we do it that way rather than just letting anyone submit to the contest is for safety reasons. So what we don't want people in their kitchens, right? Culturing things they don't know what they are and potentially getting into trouble. So what we've done is partner with these labs. They are set up to have the public come in and work with transgenic bacteria. I mean, they do these kinds of things all the time. And so they were the natural partners, basically, for this kind of thing. So we had, this past year, we had 10 community labs around the country do these workshops. And some of them had kids come in. Some of them weren't set up for that. But we did have a number of labs where they had kids come in and participate in the workshops. And that's where these are coming from. Yeah.
What cities are those in?
Oh, goodness.
Birmingham, Alabama, maybe?
We can talk about that. No, they tended to be on the coasts because that's where these community labs are mostly set up, right? So we actually had 3 different sites in New York City, which is great for New York City, right? But maybe not ideal for everyone else out there. But we're hoping to expand the program. There are definitely community labs that exist that I know about that we haven't partnered with, and we're hoping to tap some other resources as well. So,
All right, so we have about 7 minutes left. I want to say to the audience in the studio, if you have any questions, feel free to raise your hand. We'll get you a microphone and you can ask a question to our panelists. If you're watching this on YouTube, please feel free to leave a comment, ask some questions, and hopefully we'll all be able to learn together. And as an added bonus to people in the studio, if you ask a question, we'll toss you one of these fuzzy microbes here. All right.
yeah, there you go.
All right, so in the meantime, while I'm letting you incubate some questions, we've talked a lot about agar art in this session, right? But I've been walking around the microbe meeting, and I've noticed that there's a lot of microbial art at this meeting. There's stuff for sale, there's talks being given on microbial art, and it's not all agar art, right? Mm-hmm. What other forms can microbial art take?
So I think something that's gaining momentum is using the pigments to put on other things. So Anna Demetriou, she makes like quilts and puts microbial pigmentation, sort of extracts it and uses it as a color source without like all the ethics involved in using live bacteria. So I think that's one way of doing it.
Yeah, so I think people don't necessarily know like that this has been going on for like a long time too. So there's actually one of my favorite kinds of microbial art, that these antique microscope slides where artists used to arrange diatoms, which are microscopic algae with basically glass shells. So they would digest away all the organic stuff and then very carefully arrange the diatoms on slides in these like fascinating geometrical designs, and then fix them there so that you could mount them and look at them later on. I mean, these things are amazing. You should Google diatom arrangement art at some point.
You mentioned that it was so fine that they used camel hairs.
Yeah,
move
they had
them?
to use a waxed camel hair to arrange them. Apparently, it's the only thing that's both strong and thin enough to do the work, right? So, I guess the technique is very similar to how they used to fix watches. You know, the kinds of Swiss watchmaking things that went on in the 18th and 19th centuries. But yeah.
Wow. And Katherine, have you seen any kind of microbial art outside of the Agri Art exhibits that got you excited?
I have. I mean, again, being someone who's maybe not as up on the art scene, I love science-related paintings and that sort of thing. I love anything that's got a little clever twist to it, where they're taking advantage of some property of the organism that they're depicting, right? Like we were talking about with the agar art, with the breast and the fetus, right? Same idea, but with painting or something like that.
That reminds me, there's this piece I love by an artist, Michelle Banks, who's actually here at the conference, where she looked at the sequence of the microbes that were present on smartphones, on people's smartphones, and then she made a painting where she depicted a smartphone that had the bacteria isolated and sequenced from that smartphone on the image of the smartphone. So I really liked that one. Like you said, it kind of combines the realistic, microbiome of the real world with the artistic expression, and I think that's really cool. So it looks like we have a question from the audience.
Can I throw this at you first? Cool.
Nice throw.
Hi, my name is Kimberly Acevedo. I'm currently a student at Dominican College in New York. My question is, I'm really interested in this, however, I'm not artistic at all. So I wanted to ask, would you guys recommend maybe like sterilizing your tools and maybe trace like what you're trying to draw on the agar first before you draw?
That is the only way I do this. Yes. So I draw all of my designs with pencil first, glue it into the lid of a petri dish, and put it under the dish. That way, when you pick up the bacteria, you're not tracing onto the template, but it's there under your dish. So, I don't do a lot freehand with the microbes. Everything is planned first.
Yeah, like one of the weird things is it's like drawing with invisible ink, right? Because you can't see the design as you're making it because they haven't grown yet. So,
And actually, I will say that when kids do this, they sometimes are disappointed because they can't see it right away. And so you have to say, no, just wait, give it 48 hours, and when they come back in and see their designs, then they're really excited.
Or I've also taken designs from the internet and traced them. It's as easy as tracing. It's not as hard as drawing.
All right, we have another question over here.
Hi, Mandy Pagel. I'm from Wisconsin. And oddly enough, I have an art degree in ceramics and a microbiology degree.
All right.
Represent.
So my question is, are you using straight cultures or are you using dilutions when you're painting or drawing?
well, that's something that we struggle with. What the best thing is. So with the students, they're usually going from a petri dish to another petri dish, like working from growth on a plate and going back to another one. We've experimented before with using liquid cultures or concentrating the liquid cultures. I think, it depends on the organism, really, I think. And if you were really wanting to master a particular palette, you could figure all that stuff out. I think.
It really comes to knowing the organisms you're working with.
Yeah, and I would say it also depends on the type of effect that you're looking for. So there was a piece that was submitted in the first year, 2015, of the contest, where, and it was from a woman in the UK, and what she did was cut out these intricate designs in filter paper, and then soak it right in the suspension, and then put it on the plate, right? And I mean, she got amazing pieces from that. So.
All right, so unfortunately we're out of time right now. This has been a fascinating discussion, and thank you for our audience questions. Thank you all for joining us today. The work you do is amazing, and the Agri Art Competition is outstanding, and so definitely check that out if you haven't yet. And if you're watching on YouTube, make sure you go to the ASM Facebook page and check out some of those Agri Art entries. They're fascinating to look at. Thanks for joining us.